Hello and welcome back again. This is Ms. Bushra with you with our last video for this short series for exam preparation for Wizari 2025. Now, today we're marking our last two chapters, chapter 7 and 8, and we're going to continue with the same energy, strategizing and planning how to answer each of these questions. I hope you watched all of the videos, and let's continue with question 56. See, it says a plant, okay, let me get the pen. A plant with the genotype A capital, A small, remember, this is a genotype because we got the letters, A capital, A small, produces two types of gametes. Gametes means sperm or egg or the sex cells, got it? So, one got A. Among them, we created two gametes, okay? One got A capital, the other one got A small. What does this tell us about the behavior of this genetic distribution? If you remember in chapter 7, section 1, Mendel created two laws, law of segregation and law of independent assortment. However, law of segregation talks about allele separation. These two are alleles of the same gene, each one of them. And alleles are alternative forms, okay, of the same gene. Since they are talking about the height, for example, each one of them is the height, but one of them probably short, the other one probably tall. It applies on every other part of the flower, plant, or any organism, not only plant. Good. As for law of independent assortment, why I know that this is not about it, it's because in law of independent assortment, we're talking about the genes located on the same chromosome but far away, and how are they going to change or behave, or they get separated, or they get closer, or they are inherited together, okay, during these gamete formations. Here we're talking about the segregation law, which says two alleles get separated during meiosis and creation of gametes. So let's see which of these statements match the law of segregation. Let me write it. Law of segregation. The word segregation means separation itself. So we're looking for that word. Dominant alleles are passed more often than recessive one. This statement is not even correct. Not only for this question, but it's not even correct entirely. Dominant traits could be more often seen, but as for alleles, they're equal in the number of distribution, okay? Good, so this is out. Each gamete receives both A capital and A small. We see it in the question, it says each one of them got one. So they are not both inherited at the same time. And this is not even correct. Alleles get separated. This is out. Question or like sentence C says the two alleles separate, segregation separate, so that each gamete gets only one, just like how we said it here, separate from the segregation and only one, like the sentence of the question said it itself. We're holding on to this answer. Most likely this is the answer. Gametes are randomly made without using allele. Okay. That statement is not even found in the genetic. So we're going to skip this out, and our answer was C. Okay, we're doing great until now. Let's move on to question 57. Try to screenshot it and solve it yourself before you look at what I'm going to do. So it says Mendel used self-pollination for producing. Okay, self-pollination is when, okay, a flower, let's say this is a flower, okay, it is going to pollinate itself. Either the same flower have both male and female gamete, or flower on the same plant. They have the same root and they're the same genetic, so they could give it back to this one. It's still the same, it's still self-pollination. As for this flower, if it is going to send its pollen to another different flower, this is called cross pollination. This was self-pollination, self. Got it? That's first. Let's hold on to this information. All right. For Mendel created two pure breeding parents of different traits. One of them dominant, the other one recessive, both of them homozygous. They created F1 generation. Okay, why did I do that? F1 generation and then F2 generation. Pure breeding, in order to bring pure breeding, we usually try to make sure that these plants are always reproduced among themselves, self-pollination, in order to get pure breeding, or else you don't get it. Okay, so we got these two by what? Self-pollination. Good. What if we want to try the combination of these two, this cross? We get F1. This was cross pollination. Got it? Good. As for F2, we want to see what F1 got from these two parents. And in order for us to check it with F2, for tracking inheritance and how they work, self-pollination is a must. So for F2, we again make F1 do self-pollination. This is how Mendel worked. Again, P and F2, we got these results based on what? Self-pollination. So anything with F1 is out. F1, 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 F2, and P, we got the answer. Answer is C again. This was a very easy question. It's recalling from memory since it's in the book, and the process is always the same. Mendel experiment never changed. Two parents, pure breeding, self-pollination. Then by cross-pollination between these two, we get F1. Then by self-pollination of the F1 offspring, every F1, we get F2 offsprings, okay, the third generation. For phenotype ratio, okay, the first thing I want you to know, this ratio, you see it in multiple, places throughout the book, but for phenotype, it's a bit more specific. We can clearly see this in genotype too, but we are going to discard that part. It says, where can we find 1:1 ratio in a monohybrid? Means the table is, 2 by 2. Okay? Why and why? Let's see. This means we have phenotype 1, phenotype 2, like the parents, and phenotype 3 is made. Usually what we learn in the dominant recessive combination, that if one parent have a phenotype and the other one have the second phenotype, the offspring have either this or that. This is dominant, complete dominancy, okay? So we're going to take this out because we cannot have 3 phenotypes in complete dominance. You could try it. Try again and again, and you could never see it. As for incomplete and codominance, it's a totally different story, all right? It's correct for both of them, but let me see what is the difference between these before answering. If I have, for instance, okay, the incomplete dominance, it's written here, but I'm going to put it there. In incomplete dominance, you get the traits like that. If you have red, I'm going to explain it very simply with the colors. If you have red and blue, and you combine them, you get purple. That's what incomplete says. There will be an intermediate or mix or blend. As for the codominance, it's the same thing, okay? You're adding 2 color, it gives you the third trait, but not in the same meaning, okay? Not in the same meaning. These two are appearing together without blending. No blending involved. Let me give you like just a visual representation of what I'm saying. Didn't both of them appear? Yes, I see blue, I see red. They are not blended. Just like a flower, the 4 o'clock gives us pink from red and white. But there are other flowers, some of the roses. The red is lines among white lines. So it's going to be 1 line red, 1 line white. There's no blending. And it's the third phenotype. We cannot consider it as the other two, just like the other two. The offspring have a new phenotype created from the mixture of both parents. So this can be seen in the phenotype of these two. As for genotype, it's common even in the complete dominance, especially when we're crossing two heterozygous. Good question. Let's go to 59. If two dominant trait parents are crossed, all right, two dominant trait, it did not say genotype. It could be any combination. That's a bit confusing, but we have to prepare ourselves. If they're crosses, chances of getting the following, all right, this is confusing because it's not telling us about what genotypes are the parents. So we have to assume, what if both of them, let's use R since it's used R. Okay, what if both of them are R capital, R capital? What if one of them is R capital, the other one R capital, R small? And what if both of them are heterozygous? We cannot do any homozygous recessive with them because it says both of them are a dominant trait. You see, in this situation, since all of them are the same gamete, 100% is possible to get R capital, R capital. This says that A is correct. For now, this is correct. As for R capital, and R capital, R small, you will get 50% the chance of getting R capital, and 50% the chance. You could even use Punnett square to make sure that this is the right ratio. 50% of this one. We don't have any problem. For the last one, the heterozygous, 25% R capital, 25%. Okay. 25% are capital, are small, and let's see, this was not correct, are small, and 50% are capital, are small. So what it tells us here that there's these three possibilities for getting R capital. One of them is 100%. For R small, it's impossible to get R small 100% from two parents that have dominant trait, because dominant trait, when they share it, and if you use the Punnett square, the maximum amount of R small we get is 25%. Okay, that's the only amount from two dominant parents. In these two, it's 0%. Okay, as for R capital, R small, if you can tell and you see the pattern, it's always 50%. Okay, it's always 50%. Do we see any 50% here? No. So this is not correct. This is not correct. We cannot choose these two. A is the correct answer for this question. Don't get confused. Don't get panicked when you see something like that. Get your probabilities and possibilities in line. Prepare them like that, or even use Punnett square. You have time. 3 hours is enough for you to try all of these. I could recall it from memory. You could use Punnett square. Okay, let's move on to our last question from this chapter. It says, if two true breeding parents, okay, true breeding means pure, okay, pure. When you say pure, it could be homozygous recessive or homozygous dominant. It doesn't matter, all right? We don't know yet. Is it dominant or recessive? Let's figure it out. Parents are crossed with each other and all the offspring have round seeds like their mother. This is a very important point here. When it says all offspring, this happens only when you have dominant trait. Because if a dominant trait is not there, not all of them could have this. We cannot say this absolutely. So they are what? Dominant true breeding. Basha? The offsprings have round seeds, okay, for round seeds in the mother's side. Round seed, true breeding is like this. It should be homozygous, 100%. It did not, since it's like a dihybrid, it did not say what is the color of the seed, but we're sure that the mother have round seeds. Good. And yellow seeds like their father. Okay. We did not talk about the texture of the seed from the father side, but all of them got yellow seed because of the father. So Y capital, Y capital. It doesn't matter what is here and what is here complementing. As long as the mother have R capital, and the dad have Y capital, Y capital. In this table, you could see clearly we have this combination for the dad. There's R capital, Y capital, Y capital. Okay, that's a good one. R small, R, Y capital, Y capital. We cannot choose these two because they were recessive genes. And here we said true breeding. It cannot happen. So C is out just by finding out about the father's genotype. Good. Let's see if one of these two, okay, is crossed with the female that has R capital, R capital. This is our answer. This is R small, this is R capital, R capital. We're going to choose B because the female could not have R small. This is our answer. I hope you got it. It's a very nice question. It's a new style, but you can take information, sort it, plan it, organize it. Everything is going to be easy afterwards. We're at chapter 8. We're starting with question 61. It's about the nucleotide and how they're tied together. Okay? There is like two things about DNA. One is the bond between two nucleotides on the same strands on the side. The other one is between the base pair in the middle, okay, which holds both strands together. If we draw a DNA, I'm going to make it as simple as possible, okay? Let's not give it too much. Let's assume that this DNA has this many nucleotides, okay? And these nucleotides have their own shaped or own nitrogen because the nitrogens are changing. And then let's draw more of them with different color, okay? Like this. Okay, the thing here is that these nitrogen bases are bonded together, okay? Either with 3 bonds or 2 bonds, like these two. The 2 bonds are A and T, and the 3 bonds are G and C. So let's assume that these are like the C and G, and this is A, and this is T. Good. In between, closing these two ladders is with this type of bond that is horizontally like that. As for the sugar and phosphate on this sides, two sides, they are also bounded together because this is one section and one section. How do they get together? Getting these two nucleotides together is with covalent bond. As for the nitrogenous bases, either 2 hydrogen bonds between A and T or 3 hydrogen bonds between G and C. Let's not forget it, okay? So ionic bond is out of question for both of them. For nucleotides, the keyword is this, on the same strand. This means this, okay? It is covalent bond. All right, not hydrogen bond. As for between nitrogens, bases on opposite strands, this is on this strand, this is on the other one, they get together by hydrogen bond. And let's not forget that C and G are connected with 3 hydrogen bonds. As for A and T, they're connected through 2 hydrogen bonds. That was easy and simple, just as a reminder. That was warming up. Good. As for solving questions like that where numbers are involved, you don't need to know much. There's just certain keywords. Please don't forget to highlight them in the exam. No matter how much you know, if you don't put it into circle, don't underline it. Circle, you will forget about it. So let's do that. It says if the percentage, so we're asking for percentage, of nitrogen bases, all right, in one strand. This is a very important keyword. If it says sections of DNA, if it says double strand, if it says 2 strands, or it says like a whole DNA molecule, all of them have the same meaning. But when it comes to one strand, things are going to change. Let's see why. And we have 2 questions to explain both of them together. On one strand, the repetition of each nitrogen bases are not consistent and they're not equal, all right? It's only complementary to each other when we have two strands. We're going to put it into action. Let's assume that, okay, this is the one strand. It says it has 15% A. All right, good. A is a purine. And of course, G is 10%. G is also a purine. And since we are asking for nitrogen, for the percentage of the purines and pyrimidines against each other, we need to add G plus A together to get the answer. 15 plus 10 equals to 25%. This is the number of purines, 25%. As for the rest of it, T is 40% and the, where is it? The C is 35%. We're going to add C plus T for pyrimidines. It is equal to 35 plus 40. We amount for 75% only on one strand. So pyrimidine is 70, let's see, 75%. Good. See, if we divide these two by 25, this becomes 1 and this is becoming 3. This is the ratio of the purine to pyrimidine. Do we find this here? Yes, that's how we got our answer. If it says one strand, as for double strand and it asks for the purine and pyrimidine, nothing else, purine and pyrimidine, it is a totally different concept. Why? Because the strands are complementary. Let's say we have CCA, okay? That's 3 pyrimidine to 1 purine. One strand is like that. But since the other strand is complementing it with the opposite, if we have pyrimidine, it becomes purine and so on, we will get something like that. And there's T. 4, 3 here and 1 here pyrimidine, and 3 here and 1 here purine. This is 4 by 4. So in double strand, in whole molecule or one section of DNA, okay, a whole molecule of DNA or one section of DNA, it's always equal amount of purine and pyrimidine, even if we didn't try to solve it. I could choose this one immediately without anything, but let's try to solve it. CA and G are purines, and let's add them together. 30.4% plus 19.6% equals to 50%. And same goes for the other one. 19.9 plus 30.1 equals to 50%. And 50-50 is 1, 1. You don't need to think about it. You don't need to add them together. Once you know it's about a DNA molecule, a section of molecule, or double-strand DNA, both of them together, it's the same answer always. But for what? For purines and pyrimidine ratio, nothing else. Good. We're heading on to something. Let's go to question 65. Question 65, it says, if we have 2 picograms of DNA, good, extracted from a certain number of muscle cells. Muscle cells, let's highlight this, let's see why we need to use it, of the human body. What is the amount of DNA could be extracted from the same number of gametes? This is gamete. Since we have gamete against muscle, muscle is a normal body cell. A normal body cell have complete number of the chromosomes. Chromosomes is a form of DNA, so it has complete. As for the gamete, it has half the number of chromosomes. Got it? That means that whatever we got from the muscle or the normal body cell, we need to make it half, and this is going to be the answer for gamete. It works on every number. It's if it's about the amount of DNA, if it's about the amount of nucleotides. Got it? So what we're gonna do in order for us to find out for gamete, if the gamete was not given, we're gonna write gamete equals to normal, or let's say amount of gamete, amount of normal body cell DNA. Got it? Divided by 2. 2 divided by 2 equals to 1, like that. You could use it on any number, 2.5, it could be 100, 50,000, whatever it is. You always need to divide it. What if, okay, write these notes for yourself because I'm going to erase it. What if instead they gave us the number for the gamete and they asked us for the normal cell? You just bring the number, okay, or let's write down DNA in normal cell equals to DNA in gamete times 2. So if we have 1 picogram of gamete, we have 2 picograms in each cell of the body. Just use this, or just guess it, or just whenever they ask you for the normal cell, time it with 2 if they give you like for example the gamete. But for example, if they say what about the same number of cell of the sperm have 2 picogram, what about like the same number of eggs, how much they got, both of them from humans? Same, because both of them are gamete. If they gave you a neuron and a muscle fiber, it's the same again. Why? Because both of them are normal cells. Only we're going to use these when we have a gamete and a normal body cell, whichever normal body cell it is. Got it? There is another plot twist. What if they say like they try to extract DNA from the red blood cells like that? Red blood cells don't have DNA. Just be careful of that. Okay, they lose their nucleus and they lose their DNA. Let's see. Question 66. It says if a DNA molecule in a body cell has 500 nitrogenous bases. This is number, okay? We have number. And 40% of them are adenine. Let's just sort our answer. Adenine is 40%. And then what does it ask from us? It asks from the percentage of cytosine. The problem here is that cytosine and adenine don't have direct relationship. Adenine usually, okay, or like always, have the same amount like the thymine. So if like the thymine or the adenine is 40%, that means thymine is also 40%. Both of them together, they take 80% of the whole amount of nitrogenous bases. Then the remaining 20%, half of it goes for the cytosine and the other half goes for guanine, because these two are also always opposite of each other and they have equal number. That means we end up with 10% here and 10% here. The answer is 10%. Be careful. What if they asked you for the number of nitrogenous base based on that? Sometimes they don't ask you for the percentage, they ask you for the number. So if they ask you for the number, what you're going to do, for instance, we have 500 and I need to find out about the cytosine. 500, just time it with 10 divided by 100. And since these two are going together, it becomes 50. 50 of these nitrogenous bases are what? Cytosine. And another 50 for guanine. What about the rest of the other two? Since both of them are taking 100, 400 remains divided by 2. Each one of them have 200 nitrogenous bases in that molecule of DNA. It's super easy. Try to practice it according to what I say. If, however, you feel you need more information regarding this, comment down below and ask for clarification, or just DM me on my TikTok. Good. Question 67. It says the enzyme that joins gaps between. Okay, we have an enzyme joins gaps. All right, that's a very interesting word. Between newly synthesized DNA. Okay, that happens. Of the opposite direction. This is important. Opposite direction of what? Of replication fork. Since DNA is usually intertwined like that, in order for it to replicate, we need to break these hydrogenous bonds, and then we usually do it section by section, creating this fork shape. This is called the fork shape. And this fork shape is, separated by the helicase, while the polymerase are working in different direction. Two polymerase come, one of them here and the other one here. One of them goes to the direction of the unwinding of the replication fork. Since the replication fork goes up, this is going up with it, and the new strands of DNA that's created is continuous. It's new, it's complementing this, and it's continuous. There's no gap. The polymerase is making sure that it's going to create it by itself. While the other one, the other one is patchy. It gets section by section. Why? Because the movement is opposite, all right? Once this is coming, it becomes like that. These, because the polymerase are, attaching and detaching and going to another section in order to complete it. This could not be repaired by the polymerase itself. It requires time. So another enzyme comes and bind them together in order for them to become a complete strand. This enzyme is called ligase, okay? We have, let me change the color. We have helicase. Helicase comes from the helical shape, double helix, and it's unwinding DNA. How? Breaking hydrogen bonds between the nitrogen bases. Basha? As for the other one, polymerase, it creates polymer. Polymer yani very long strands of the same repetitive sections. So the nucleotides are the repetitive sections, and when we create a strand, we create polymer. That's why we call this polymerase. Which polymerase? DNA polymerase, because we also have RNA polymerase. These usually main function is adding nucleotide. تمام. As for ligase, بتسبب ارتام بيتوا بلزقة ligase لزقة. Sedakaten joins the gap of opposite direction creation of the DNA strand. Got it? So who is doing that? Like making sure that the gaps are joined? The ligase. لزقة. تمام? One advice is repeat any part that you find it confusing. Try to do it by yourself, and you have like past papers. Use them to apply these rules on them. I'm sure you're going to do great. Let's do question 68. Which of the following is not a function of DNA polymerase? We just now revealed one answer, is that the DNA polymerase is actually adding complementary nucleotides. So this is right. That means we cannot choose that, because we're saying we need something that is not about the DNA polymerase. Another thing, it's finishing DNA replication. Yes, the DNA polymer is the one that creates the new strands, and it is going to seal the strand that is going to the direction of the replication fork. And it's also terminating the whole process. So this is also right. Along with doing that, it's also like a checkpoint, checking every part to see whether there is any error. So it's also doing proofreading, okay? Proofreading is checking it again, like a checkpoint. These are all correct about polymerase. So we should have something in the end that is not about polymerase. It says breaking hydrogen bonds between the DNA. We said that this is the job of helicase, okay? Very simply. That's why we're going to choose D as our answer, because it's not a function of polymerase, but a helicase. Good. Very simple question, but please memorize these three functions. Most students think that the DNA polymerase is only adding nucleotide, but it's actually doing more than that. Let's see. It says DNA and RNA are similar in which category here? See, DNA and RNA, they have so many differences. The ones that we can say that from structure based, the ones we know about it is that DNA is double helix and 2 strands, while RNA is 1 strand and much shorter. The length of one gene, it could be in 3 forms too. As for the chemicals inside it, how they're made, the nitrogenous bases of DNA, okay, the DNA and the RNA are different. How? The nitrogenous bases that are created in DNA is A, T, G, C. Here in the RNA is A, U, this is the difference, G, C. So they are not similar in the nitrogenous bases. As for the sugar backbone, no, they don't look like each other. With the proof, DNA itself, the word stands for deoxyribonucleic acid. Got it? Deoxy here, deoxy, it's like making it a negative form, means there's something lacking. Oxy means oxygen, so we're lacking an oxygen in ribonucleic. This is the sugar that makes up the DNA. Okay, it's a nucleic acid, that's why we add this suffix to it. So it's a sugar called ribose, okay, but it lacks 1 oxygen than the normal other ones. As for the RNA, RNA stands for ribonucleic acid. This means that sugar doesn't have any problem with the oxygen amount. It has a complete amount of oxygen. So they're not the same in the sugar as well. Okay? What about the phosphate group? Phosphate group in both of them is PO4. This is the only similarity between them here. So we're choosing C, we're crossing these out. Phosphate group is the similar chemical features between the DNA and the RNA. Good question. Simple, but good. As for this one, it says double strand regions on RNA. Okay, we said RNA has single strand, but there's a plot twist. RNA is simple strand like that, but sometimes, just like how ropes or like threads, the 2 ends are coming back together, creating like a twist here, like this. Okay, and we have the nucleotides here. على أساس nucleotide هندرس كردينا. When they get close enough, if there is an A and a T close by, they create a bond between each other. And if there is a G and C, they create a bond to each other. This is telling us something, because we're looking for the bond. Just like DNA, the backbone here, okay, is made of covalent bond. Covalent bond. Good. So normal RNA that is linear have only covalent bond. Let's not forget normal RNA. Write this for yourself. But RNA, where they have regions of double strand, like this region here, create hydrogen bonds between the nitrogenous bases, these hydrogen bonds, okay? So in this area, what do we have? Both covalent here on the side and the hydrogen bonds. Hydrogen bonds. And since we're talking about the double strand region, we're going to say both A and B covalent and hydrogen ionic. Every single time you see it, just throw it away because we don't have it. I usually tell my students the vertical is covalent, the horizontal between the nitrogen base is telling you about the hydrogen bonds. Good. We're on to our last two questions. Which of the following is a start codon? You probably say, oh, why Ms. Bushra put something that easy? We're just memorizing it. Yeah, you're memorizing it, but when you're stressed about something, you usually make mistakes, like silly mistakes, right? So here I'm going to give you something that helps you memorize it. We all know that AUG stands for, sorry about that, stands for what? For the start codon. I could choose it clearly like this. But the other stop codons, you might confuse it or you might forget about it, and there are three. I had a way to memorize it back in school. I'm going to teach you. Making sentences that you're going to memorize the whole sentence will remind you of these. Let's see how. I was using these and say, you, okay, are awful. Why? Let's see why. Because you always gossip. See, gossiping is bad. People will hate it and get away from you. That's why they say you go away. So apparently these are two friends and one of them says you are awful because you're gossiping, so go away from me, right? Each one of these initials, UAA, UAG, and UGA. It's easy for some students to memorize it quickly, but for others, they need like an extra spiciness to it, just like this. So I hope you're going to use it and if you find it useful. Question 72 is quite frequent in the Wizari questions, and this is from 2024 first trial. I brought the same question. We need to read it carefully in order for us to understand it. Before I start anything here, let me tell you how you're going to do it, okay? Let's assume that we have, okay, a DNA that's unwinded for RNA transcription. That side of the DNA, or that strand of the DNA, that transcription happens here, we call it template. And since both strands are complementary to each other, we call this complementary strand. Got it? So we have template where transcription happens, and we have complementary strand. If they gave us complementary strand, we have to make it into a template, then change it to the codon, or else it doesn't work. So we're having this, and then we also have the codons from mRNA. We have codon, all right? And then we have tRNA that's gonna bring the amino acids for us. It has anticodon on them. And lastly, the amino acid. Got it? Lastly, we have the amino acid. So let's assume that we have ATA. Got it? ATA. What if they gave us ATA here? We have to like make it into the template, then we're gonna do it. But the complementary strand here is not needed since they gave us the template immediately. Only if they give us TAT, then we're going to change it to this and then we're going to move on. It's always like that. Let's change color. ATA in the mRNA is going to be changed to UAU. Why? Because this here on the we have like the opposite pyrimidine on the other side of the RNA doesn't have T. We're going to switch it to U. And T here, just normally like any other time, is just going to give us A on the other side. Good, that's the codon. Then for the anticodon, we're going to like flip it another time. U becomes A, and we're still at RNA. A becomes U, and U becomes A. Got it? AUA. This is the anticodon. And based on the codon, we don't always need anticodon, but reading amino acid is usually based on the codon. Sometimes, like the question I gave you in the Wizari, they give you a table for anticodon instead of the codon. We can use it and we can change it here. According to the requirement, we're going to change it. So let's see. We always start with template, codon, anticodon, and amino acid. If there is no anticodon in the table, it's only codon, we can skip this part too. Good. So once I have this, okay, the complementary strand of the template, I need to change it to the template immediately because this is not going to work for the codon. Since it's DNA and we're going to change it to another DNA, we don't need to use U. T becomes A becomes T, and like that, so on. Let's do it. GTT, okay, AG, A, and C, this is AA, like that, and then GTA. Now we can work on it and make it into a codon. Why? Because we found the template. It's complementary to this strand. Now, if you change this one to the codon, this is the first thing we should do. You cannot go to the anticodon immediately. Even if you're like smart about it and say you could do this, don't do it. Don't risk it. Anything is risky in Wizari, don't do it, please. No hacks, no tips, nothing. All right, let's change this. A's are changing to U. U. T is changing to A. So U, A, U. That's the first one. Good. GTA, we are going to make it C, A, A. And then the AGA, U, C, U. CAA, we're going to make it G, okay, U, U. And then the last one, GTA, becomes C, GTA, yeah? Okay. It becomes CAU, like that. Okay, we have the codon. Then because the template here, okay, sometimes stops here, here it asks us in the table anticodon. We have to change this to the anticodon before we start anything else. Let's change this to blue again, and we're gonna flip it again. We're still using U. UAU becomes AUA. Good. And then CAA becomes GUU. UCU becomes AGA. And then GUU becomes CAA, and CAU becomes GUA. Just like that. Okay? GUA. Now we have the sequence of anticodon. What is this? Anticodon. We have the anticodon. Let's check it out. The anticodon AUA, the first one, where's AUA, is tyrosine. We got it. So the first one should be tyrosine. Histidine is out. Good. See? Tyrosine. And then GUU, there you go, glutamine. Good. So we have this, we have that. Okay, glutamine. And then, is it histidine or serine? Between these two, we're going to find out which was the answer. Since we have AGA, and AGA stands for serine, it means that D is the answer. We could check the valine as well. CAA is valine, histidine is GUA, and we have it here. We use the anticodon to find the answer, and it was correct. If the table, however, here was written codon, we could have stopped here and just got the answer like that. I hope you got everything right here. You understood everything from this, type of questions. And this is the mark of our last video, last part, last question. I hope you're gonna have a great experience in the Wizari. Best of luck. Thank you.